Hello, my name is Ann Moser. I'm at the Peroxisomal Lab at the Hugo Moser Research Institute at the Kennedy Krieger Institute in Baltimore. I'm going to describe the measurement of phytanic acid, phytol fatty esters, and free phytol in foods that we have been doing for the DARE Foundation. Refsum individuals lack the enzyme to alpha oxidize or break down phytanic acid. Humans cannot synthesize phytanic acid except from precursors, the phytol fatty acid esters and free phytol. However, phytanic acid and its precursors are present in certain foods, and thus phytanic acid accumulates in blood, tissues, the retina, and fat stores. If the level of phytanic acid is very high, neurological symptoms can occur. And retinitis pigmentosa is often the diagnostic symptom for adult Refsum and can progress for years, leading to light blindness and loss of vision. The first dietary therapy for Refsum was established more than 50 years ago by Doctors Refsum and Steinberg. The restriction of foods that had a high content of phytanic acid resulted in lower plasma phytanic acid and improvement in neurological status. The new finding from Doctors Krauss and Vetter that phytol fatty acid esters are present in foods and can be converted to phytanic acid by human digestion is an important finding for Refsum. Now we recommend a diet which is low in phytanic acid, phytol fatty acid esters, and free phytol. Acute weight loss should also be avoided as phytanic acid is released from fat stores and can be toxic to the nervous system. The DARE Foundation has funded the testing of phytanic acid, the phytol fatty acid esters, and free phytol in foods. This slide shows the bioavailability of esterified phytol and its relevance to Refsum disease. Here is phytol attached to chlorophyll, and it stays attached to chlorophyll except if it is digested by ruminants such as cows and sheep, and where in the second stomach of the cows it is converted to phytanic acid, and thus phytanic acid is in the fat of the cows, such as in butter fat, milk fat, meat fat. And marine plankton can also metabolize the phytol from chlorophyll. Thus, phytanic acid is high in oily fish from the ocean. Chlorophyll is also present in fruit and vegetables. However, the human digestive system cannot metabolize the phytol, cannot cleave this phytol from chlorophyll. However, during fruit ripening and food senescence, the chlorophyll is broken down, such as you can see the leaves turning from green to red during the autumn. And these leaves, if one were to eat, the leaves would be high in the phytol fatty acid esters. And the human digestive system can metabolize the phytol fatty acid esters to free phytol. And then the free phytol is converted also by the human digestive system into phytanic acid. Thus, this foods that are high in the phytol fatty acid esters may be a problem for Refsum disease. Stephanie Krause measured fruits and vegetables and tested for phytyl fatty acid esters. On the group on the left, she found no or low PFAE, and the foods on the right, including nuts, legumes, lettuce, she found significant amounts of phytyl fatty acid esters. So here is the DARE food list that we tested last year, and it includes foods from all different types of foods: meat, fish, nuts, seeds, grains, oils, shellfish, legumes, fruit, vegetables, fermented foods such as yogurts, cheeses, and dairy. And there are also foods that we will be testing this next year. The food is purchased by me at the local supermarket, and if it's unavailable, I order it online from Amazon.com or other food vendors. The following steps are done in my home kitchen. I prepare the food for eating. The vegetables are steamed, the meat broiled, excess fat or water is blotted and removed. Then the food portion is weighed. The foods are minced and stored frozen. The minced food is taken to the peroxisomal lab at the Kennedy Krieger Institute. In the lab, I weigh a food portion. Here I am in the weighing room. Usually about a gram is analyzed, and the extraction of the food lipid is done with the chlorphromethanol solution. Then the total lipids are weighed, and a portion of the total lipids are taken for total phytanic acid, and the phytanic acid is measured by Ann Snowden and David Jones. And another portion of the total lipid extract is taken for total phytol analysis. And this is analyzed by GCMS, by Kirsten McGee here. And then another weighed portion is taken for analysis of the phytyl fatty acid esters. The phytyl fatty acid esters are purified by silicic acid column chromatography. And here is a column showing the separation of the phytyl fatty acid esters from chlorophyll. You can see the chlorophyll band. This happens to be spinach, so it has a lot of chlorophyll. And the chlorophyll band remains at the top of the column until fraction 4. Thus, the fraction 1 contains the hydrocarbons, fraction 2 contains the phytyl fatty acid esters, fraction 3 contains the free phytol, and then finally the fraction 4. So after alkaline methanolysis, we can measure the phytol in all of these different 3 fractions, and we measure that by GCMS. The results of the food analysis are given to the expert nutritionists, and our expert nutritionists are at the Refsum Clinic in London, and these nutritionists are also on the board of the DARE Foundation. They are Eleanor Baldwin, Radha Mashadran, and Sarah Furman. And these nutritionists have established a traffic light system for the Refsum dietary recommendation.